How do enzymes work? So just like what Haldane mentioned in his book Enzymes back in the 1930s, your substrate should bind to your enzyme. And because we know that enzymes have a specific active site in which the reaction takes place, your substrate must bind on the active site of your enzyme. Now, through years of studying the structure of enzymes together with their substrates, there are some general observations. Number one, in this so-called enzyme substrate complex, we see geometric complementarity between the active site and the substrate. This means that the general shape of the active site is complementary to the shape of your substrate. So if we see some aromatic rings, for example, in our substrate, then there should be some space in our active site that would allow these aromatic rings to enter the active site. In addition to this, electronic complementarity is also observed. This means that if you have positive charge on the surface of your substrate facing the active site, these positive charges are met by negatively charged groups found in the active site, and vice versa. So this geometric complementarity and electronic complementarity allows the substrate to perfectly interact with the enzyme. These have also been the basis of the lock and key model. So the lock and key model survived for long years. So it's the model of how your substrate interacts with your enzyme. So just like how your key is complementary to the lock, your substrate is complementary to the active site in the enzyme. But through the years, this lock and key model was revised. So if we include the fact that your molecules, the atoms in your molecules are not rigid, so they are actually flexible, they are moving in real time. There is what we call the induced fit model. So in the induced fit model, we don't immediately see the complementarity, especially the geometric complementarity between the substrate and the active site of the enzyme. So the binding of the substrate itself induces minor conformational changes in the enzyme and also in the substrate. And after these conformational changes, that's when we see the perfect complementarity of the substrate and the active site. That's the induced fit model. In recent years, some scientists have shifted their focus to what is called intrinsically disordered proteins, or commonly known as IDPs. So some enzymes, actually it's just only a small number have been discovered. Some enzymes are IDPs. So in their native state, they don't have a specific three-dimensional structure. So they are just like polypeptide chains floating inside the cell. But if they encounter their substrate, that's when they take a specific three-dimensional structure. So this is something new. So it's not aligned with the lock and key and the induced fit models. Now, some additional comments on the interaction of your substrate and your enzyme. So the interaction must not be too strong because if the substrate is bound tightly to your active site, then chances are, yes, the reaction will take place, but the product might not leave the active site at all because of the strong interactions. On the other hand, if you have weak interactions, very weak interactions between your substrate and the active site, then your substrate can leave the active site even before the reaction has taken place. So enzymes and substrates, they were optimized to have this perfect degree of interaction. Now, in the mechanism of a specific enzyme, their stereospecificity is important. So enzymes are stereospecific. And this is because they are inherently stereospecific. So if you recall, only L amino acids are used in the synthesis of proteins, including your enzymes. So we have not seen any protein made from D amino acids. And because of this stereospecificity of your enzymes, they bind to one specific stereoisomer. So for example, let's go back to aconitase. Aconitase catalyzes the isomerization of citrate to isocitrate. This is one step in your Krebs cycle. If you notice, citrate is prochiral. Means if we replace one of the carboxymethyl groups with another functional group, the molecule becomes chiral, which is the case in this reaction. Now again, your aconitase binds only one stereoisomer, or actually, since citrate is prochiral, it will bind citrate in a specific manner. So one of the carboxymethyl groups is inside the active site, while the other carboxymethyl group is sticking out of the active site, meaning only the carboxymethyl group that is in the active site is the one that gets the hydroxyl functional group, and not the carboxymethyl group sticking out of your active site. So it's not this one, but it's always this carboxymethyl group that gets functionalized. Enzymes also vary in their geometric specificity. So some enzymes bind only one substrate. Some enzymes can bind two or more related molecules, like your alcohol dehydrogenase. This is the enzyme that can catalyze the conversion of methanol and ethanol into their corresponding aldehyde. Now, this is actually the principle behind the first aid to methanol poisoning. So they say if you are exposed to methanol or if you ingested some ethanol, you can prevent any further damage by ingesting ethanol. And this is because your alcohol dehydrogenase acts on methanol, transforming it into formaldehyde. This formaldehyde, you know what formaldehyde is, right? It's the one used in preserving um, stuff. And this can cause blindness. Now, since ethanol has a bit of higher affinity to alcohol dehydrogenase than methanol, ethanol can replace methanol in the active site of alcohol dehydrogenase. So, instead of your methanol being converted to formaldehyde, you have ethanol being converted into a um, safer compound, acetaldehyde. Now, other enzymes, they have a wider spectrum of substrates. The example would be your digestive enzymes, like your pepsin, trypsin, and so on. They can act on almost anything that they encounter in our stomach fluid.